Hello and welcome to another edition of Zog Science. Today we're going to be taking a look at classification and the system for how we classify organisms. So a little bit of history. The study of classifying organisms that is known as taxonomy. And Carl Linnaeus was the first person to come up with a comprehensive system. First of all, he came up with the idea of binomial nomenclature. So essentially at the time you have a lot of people who are calling different organisms by their regional names. So, someone in Europe calls something one thing, like calls a squirrel by their, the name for their home language. Somebody from America calls a name by their language. So Carl Linnaeus comes up with this system that puts it all together, and that is that you have the genus and the species. The genus and the species is always italicized when it is written out, and the genus is always, first letter is always capitalized, the species is lowercase. He also came up with the idea for a hierarchical classification with kingdom, phylum, class, order, family, then genus, and species. Now, when he first started off, he just only had two kingdoms. He had animals and vegetables. Those were his two kingdoms, and then everything else fell into that. And we have since built upon these systems and improved them. We'll talk about that today. But some problems with the old system. Not necessarily all of them have been fixed, although a lot of them have. The old system was based largely on morphology. Now we tend to base our system almost exclusively on DNA. And not every taxon on the same level is equivalent. So what that means is that a kingdom, or not necessarily a kingdom, but like a family in one group might have a certain number of organisms that have a lot of differences, and then a family in another group might only have a few organisms that are very actually close to one another. They don't have very many differences. The number of kingdoms changed a lot since Carl came out with his first one. So Linnaeus's, again, he had two kingdoms, and that increased over time and changed as people figured out, oh, there's actually a lot more things that are going on. And the final problem is that most organisms are microscopic. And so it's really hard to use morphologies to tell these things apart, especially when you don't necessarily have very good microscopes. And even when you do have good microscopes, they tend to look very, very similar. So the current system, again, is based off of the old system. It uses DNA to look at phylogeny. Phylogeny just means evolutionary relationships, and is much more accurate as well as more specific. So this is kind of a glossing over of the order Carnivora, which has the families, or some of the families are Felidae, Mastelidae, and Canidae, and then some genuses, like we've got Felidae, and then Panthera would be the genus, and the species would be Panthera pardus, which is the jaguar, and then you can see some of the other examples. Now, in reality, there are a lot more than just kingdom, phylum, class, order, family, genus, and species. We actually start with domains now, which this domain would be Eukaryota. And then you can see down here, this is the entire full classification for Canis lupus, and you can see that there are many, many more. There are subkingdoms, there are superclasses, all these different types of things that are designed in order to make the system more accurate as well as more specific, and to group organisms based on shared characteristics. So let's talk about how we use DNA. Over time, mutations occur to each population's DNA separately, and they're going to be occurring in different, in the same genes. Different mutations are going to occur. So what we do is we compare the number of differences between different species. More differences equals a split that occurred longer ago. We can use this now to build a molecular clock. And the molecular clock is basically a way of telling us what is the rate at which a particular gene is mutating. And we can come up with an estimate of, okay, based on the number of mutations that have occurred, how long ago did this species or did this organism diverge from other species? And basically what we do is we plot the amount of divergence for known species versus the amount, how long ago those species diverged, and we can tell those things using fossil records and molecular information. And then this gives us an approximate rate of the evolution in a particular gene. And then when we find new organisms, or we take a look at new genes, we can compare them to the line that we've come up with and see whether or not they fall within the line or outside the line. This is showing mammals in a particular gene, and the three dots down here, these are primates. So it seems as though primates have a slightly different amount of rate of mutation, a much slower rate of mutation that occurs within this gene as compared to other mammals. Now, one of the problems with molecular clocks is that it's not 100% certain. There's a lot of variation. However, it does give us a good approximation for how long ago different species have diverged. Now, when we also take a look at DNA, one of the things we do is we can take a look at gene duplications. And we mentioned this a little bit earlier when we talked about gene families. But there are kind of two different types of duplications. One, or orthologous. I'm going to go with orthologous genes. And these help look at variations between species. So orthologous genes, they duplicate and they are diverged between different species. So they're the same gene, as in they code for the same trait. However, there are mutations that have occurred between those genes, and we use those to look at variations between species. Paralogous genes are ones that they are going to duplicate within a species. So instead of just having one of these genes, you now have two. And what you can have happen is that, again, there's going to be mutations that occur in each of these genes that are different. And so we can use these to look at variation that is occurring within a species and maybe even predict where some subspecies are or if a new species is potentially going to occur because you have different sets of paralogous genes within two populations. So basically, this is the current way that life is classified. We have three domains: bacteria, archaea, and eukarya. You can kind of see all the different major types of these different bacteria. Notice that the common ancestor is most closely related with bacteria. And then from there, we have archaea, which is actually more closely related with eukarya than it is with bacteria. And then our eukarya, remember that all eukaryotes have mitochondria. So the mitochondria, which used to be prokaryotes or bacteria, was somewhere incorporated along this line here. And then recall that plants are the only eukaryotes that have chloroplasts and other plastids, and so those were incorporated somewhere along this branch here. Some of the major groups within eukarya are plantae, the land plants, animalia, animals, and fungi, which is just fungi. So. Cladistics. Cladistics is a system of grouping organisms based on shared characteristics, and this is one of the ways that we would put together different groups of organisms. Usually we do this based off of shared derived characteristics, or basically characteristics that are unique to a particular group. So things like mammals having fur, or birds having feathers, things like that. Groups are called clades, and all the organisms within a group or a clade share the same characteristics. Now, a cladogram is not the same thing necessarily as a phylogenetic tree, although it can be. Because again, a cladogram is more based off of derived characteristics, whereas a phylogenetic tree is more based off of evolutionary relationships. Generally, those things are equivalent, however, not always. So here is an example of a cladogram that has been put together. You can see that the lancelet does not have any of those characteristics, and so it is considered the outgroup. It is not a part of the rest of the clade. We then have lamprey, which have the vertical column. However, they don't have any of the other characteristics. And you can see how we are splitting apart the group based on each of its characteristics. And we could consider each of these a different clade. So all of the organisms that have hinged jaws, that could be considered a clade. All of the organisms that have four walking legs, that would be considered a clade. So a clade can change based on how you kind of want to set it up. Some assumptions that we make. First one is maximum parsimony. This is basically Occam's razor. All else being equal, the simplest explanation is going to be true. So when we create a cladogram, we want it to be as simple as possible. And second is maximum likelihood. Basically, we assume that mutations rates are equal, and therefore more closely related species will have fewer differences in DNA. These are both large assumptions that we make. However, we kind of need somewhere to start when we are making a cladogram. Excuse me. So let's take a look at three species, and let's try to figure out, okay, what should these phylogenetic trees is the best? Well, the first thing we're going to do is we're going to draw out our three possible phylogenetic trees that we could have, and these are the different combinations. And we've got a gene that is 4 nucleotides wrong. What we're going to do is we're going to say, okay, looking at each of these cladograms, where is there going to need to be events occurring? Well, here's where one of the events is going to occur, right? You either have a C or an A, right? This is the ancestral sequence. So either C or A. So here's the split off from the ancestral sequence, adding the C to these two. For the other two, it's going to take two events for that to occur, all right? So already we're kind of biased towards this one, just because we can see that there's only one event that needs to occur versus two in order for this C to be introduced. And what we do is we then go through and we say, okay, let's take a look at the next ones. Where else do these events need to occur? And we just continue adding them up until we have all of the different events that need to be occurring at each site in order for each of these phylogenies to be true. And what we find is that the first phylogeny requires only 6 events, and the other two require 7, meaning that this first phylogeny is the one that is most likely true, and that would be the one that we would keep. Now, we can mitigate some of this by using more than just one sequence. And you guys saw this a little bit when we did the BLAST lab. By combining several different sequences together, we can create a cladogram that is going to have a higher probability of being correct. And generally, we use computers to do this, as you all saw when using the BLAST data. So as we do classification and as scientists are trying to classify organisms into different clades, the goal is to eventually form all monophyletic clades. And what that means is that we want all of the organisms within a group to come from a single common ancestor. That's what is known as a monophyletic group or a clade. And this is not always the case based on either historical reasons or just lack of data. We haven't gotten quite to that point yet. And there are a couple other groups that we can have. We can have a paraphyletic group. This is where you include some organisms from that are genetically related, but not all of them. Or you can have a polyphyletic group where you have organisms that actually have different common ancestors from each other. So, for example, reptiles are not considered a monophyletic group because they have not traditionally included birds. And birds, it turns out, based on DNA evidence, are descended from reptiles. And as such, they should be included in the reptile clade. But in general, we tend to think of birds as being separate from reptiles, mostly because a lot of the characteristics they have are very, very different from reptiles. So one last question to kind of wrap things up. Is this a lizard or a snake? You can kind of take a look. You see that it's legless. But however, it is actually, based on DNA evidence, a lizard and not a snake. So you can definitely get tricked out. You definitely want to be using DNA evidence when you are doing classification. All right. So thanks for watching, and I'll see you next time.